Hey y'all, Miss Mayhew here, and we are going to start going over the cell cycle and cell division. So, the first key concept that we're going to start with is 7.1, which is the different life cycles use different modes of cell reproduction. Now, when we're talking about these different life cycles that use the different modes of reproduction, we're talking about the two basic strategies for reproducing. Those are asexual reproduction and sexual reproduction. So the first type of reproduction is asexual reproduction. This happens when there is only one parent involved. So the offspring is going to be a clone, which is genetically identical to the parent. Now, because these offspring are clones of the parent, any genetic variation that occurs in the offspring is going to be caused by mutations. Okay, these mutations are changes in DNA sequences due to environmental factors or copying errors. Some examples of organisms that reproduce asexually are single-celled prokaryotes. These usually reproduce by binary fission. Single-celled eukaryotes can also reproduce asexually, but because they have a nucleus, they're going to reproduce by mitosis and cytokinesis. Okay, but it's not only single-celled organisms that can reproduce asexually. There are also multicellular eukaryotes that can reproduce by asexual means as well. Some examples of these organisms are starfish, which reproduce through fragmentation, or ivy or succulents that reproduce through propagation. The second type of reproduction is sexual reproduction. This is where you have two parents involved in making offspring. This involves the fusion of gametes, which are your sex cells. Now, when this happens, you end up with offspring that have genetic variation because they're not identical copies of one parent or the other. These gametes are formed through meiosis, which is a process of cell division that ends up creating your gametes and reduces the genetic material by half. So that way you have half from one parent, half from another parent, creating and fusing to form a whole offspring. Now, DNA in eukaryotic cells is organized into chromosomes. And if you remember back in previous units, we talked about how eukaryotic DNA is linear and it's wrapped around these histones and it goes through these layers or these levels of protein packaging in order to form these X-shaped chromosomes that you're used to seeing. And these chromosomes can be found in all of your cells. When we're talking about your somatic cells, these are the body cells that are not specialized for reproduction. So this is everything except for your gametes. Each of these somatic cells contains two sets of chromosomes, and they're found in these homologous pairs. One of the homologs comes from the female parent, and the other comes from the male parent, and each of those have corresponding genetic information. Gametes, on the other hand, only have one set of chromosomes, so they have one homolog from each pair. These gametes are considered haploid, meaning the number of chromosomes is N, or one set. Fertilization occurs when two haploid gametes fuse to form a zygote. Now, this zygote that's formed is now diploid, meaning the number of chromosomes in the zygote is 2N, or two sets. All sexual life cycles involve meiosis. Depending on the organism's life cycle, one of two things can happen. Either one, the gametes are developed immediately after meiosis, or each haploid cell may develop into a haploid organism where there is a haploid stage of the life cycle. That will eventually produce gametes through mitosis. Either way, fertilization is going to result in a zygote, and that's going to be what begins the diploid stage of the life cycle. Eukaryotic cells are able to divide and create new copies of themselves through mitosis, which is a division of the nucleus, followed by cytokinesis, the division of the cytoplasm. Reproductive signals are usually related to functions of the entire organism, not the environment of a single cell. Most cells in multicellular organisms are specialized and don't divide. Now, when we're talking about chromosomes, we're talking about a very compacted, very organized structure of our DNA. So if you look here at the top, you can see DNA in the way that you're used to seeing it, in this double helix form. You can see the rungs that are made up by those nucleotide pairs forming those nucleotide bases that are paired up, and the phosphate sugar backbone that makes up the outsides of the ladder. Now, in order to form this compact shape, that DNA is wrapped around these histones, which are special proteins, and that DNA wrapped around those histones is what forms a nucleosome. Now, those nucleosomes are going to wind around each other, where it winds up and forms what we call a 30 nanometer fiber. Now then, this 30 nanometer fiber is going to form loops that are about 300 nanometers in length. So now that fiber is looping up, and then those loops are going to be what are compressed and folded and coiled up to form that really tightly formed X shape that we talked about earlier. Now, another thing I want to point out about a chromosome is your chromosome is made up of two sister chromatids. Each of those chromatids is an identical copy of the same strand of DNA. Okay, remember, in the cell cycle, the S phase duplicates the genetic information. So what makes up that X that you're seeing here is those two copies of that DNA. Now, in mitosis, you have one nucleus from the parent cell that ends up producing two daughter nuclei, each containing the same number of chromosomes as the parent nucleus, so what it was originally. Mitosis is continuous, but it's convenient to subdivide it into phases, which you learned in freshman biology. A friendly reminder of those phases are prophase, metaphase, anaphase, and telophase. In prophase, the chromatin that is found in the cell begins to condense and form those chromosomes that we talked about previously. In metaphase, those chromosomes line up in the middle along the metaphase plate. Anaphase is where those chromosomes are pulled apart. And telophase is where we begin to form two new nuclei. The cell cycle is the period from one cell division to the next. So once we form a new cell until it divides again. We typically divide these into stages in eukaryotes. Okay, those stages can primarily be divided into two stages. The M phase, which is mitosis, and the segregation of chromosomes into two nuclei, followed by cytokinesis, which is the division of the cytoplasm. That's what ends up giving us two separate cells. Okay, whenever the cell is not actively in mitosis, we call that interphase. Okay, this is where the cell nucleus is visible and regular cell functions occur, including DNA replication. Now, that second phase, that interphase, we can actually subdivide into three subphases. G1, the gap 1 phase, which, depending on the type of cell, is variable. This could last a really long time. The S phase, which is the synthesis phase, this is where DNA is replicated, or where we synthesize a new copy of DNA. And the G2 phase is our gap 2 phase. This is where the cell begins to prepare for mitosis, where we synthesize the microtubules that are needed in mitosis that are going to connect to those chromosomes and segregate them. So when we look at the cell cycle of a cell, you can see that most of the cell's time is spent in that interphase, that G1, S, and G2. Only a small portion of the life cycle of the cell, or in the cell cycle, is spent in that mitotic phase, the M phase. We've gone ahead and included a summary table for you guys to put in your notes for the eukaryotic cell cycle that goes over all of the steps that we talked about in the previous slides, along with some photos of what is happening in each of these stages. So interphase is G1, S, and G2. This is where the cell is doing regular, normal functions. Prophase is where we have the condensing of the chromosomes, and you can see even in the picture, the real life picture at the top, you can see that they are starting to condense, and you can see their form starting to take shape. Prometaphase is kind of that weird place in between prophase and metaphase where they're still lining up, but they're not quite lined completely up yet. Metaphase is where those chromosomes are lined up on that metaphase plate. Anaphase is where those sister chromatids are pulled apart. And telophase is where we're forming the two new nuclei. So this slide is to kind of review what these chromosomes are made of and their structure, especially in relation to the cell cycle, specifically prophase, where these chromosomes are formed. So remember, in the interphase before mitosis, we made a copy of the DNA in the S phase. So there are two identical copies made of the nuclear DNA during the S phase. Each chromosome consists of those two parallel filaments, or those two copies of the same strand of DNA. We call those two things sister chromatids. Now, in prophase, the final coiling of those complex levels of organization. So we start out, remember, with the DNA that wraps around the histones, which then wraps around itself to form that 30 nanometer fiber, and so on and so forth. This is now going to be visible with light microscopes because they're starting to form those X's. Now, the final compaction enables those two sister chromatids to be pulled apart eventually in anaphase and carried to separate daughter cells without that DNA being damaged. When they are in their little X shape, they're joined at something that we call a centromere. Now, the kinetochore is going to be made up of proteins that are on either side of the centromere holding that chromosome together. Now, the reason these kinetochores are important is because this is where those kinetochore microtubules are attached to the chromosomes and what allows those sister chromatids to be separated. As you can see in this picture here, we have the chromosomes lined up along the metaphase plate where those kinetochores are attached to the kinetochore spindles. Okay, those spindle fibers are going to shorten and they're going to be what separates those sister chromatids. So those sister chromatids are separated and moved to opposite poles of the cell through two mechanisms. The first is that those kinetochores have molecular motor proteins. So these are special proteins that are able to move those chromosomes along the microtubules. The second mechanism is that the kinetochore microtubules, which are the microtubules that connect the centromere of the chromosome to the centrosome, which is the pole that the spindle fibers are coming from. Those microtubules are able to shorten from the poles that draws the chromosomes towards those poles, towards that centrosome. Once mitosis is finished and there are two nuclei for the two new daughter cells, then the cytoplasm can be split in a process called cytokinesis. Now, in animal cells, the cell membrane is going to pinch in between the nuclei, forming what we call a cleavage furrow. Okay, as that pinches, it's caused by this contractile ring of actin and myosin, which are microfilaments that form on the inner surface of the cell membrane. As that ring contracts, the two proteins are going to pinch that cell into two. So here on the left, you can see that contractile ring that's causing the cleavage furrow that happens in animal cells. Okay, this is because animal cells only have the cell membrane or the plasma membrane, which is more flexible. On the other hand, on the right side, on side B, you can see that we have a plant cell or a fungi or something that has a cell wall. Because the cell wall is more rigid, we can't just pinch it and form two new cells that way. Instead, we have to form a cell plate in between the two cells after mitosis occurs. The way that this cell plate is formed is through vesicles from the Golgi apparatus. These vesicles are going to pinch off the Golgi apparatus, and they're going to start forming along the plane of cell division where we're going to form our new cell membrane and our new cell wall. Those vesicles, as more come to that area, are going to fuse and form a new cell membrane that crosses the two daughter cells. The contents of those vesicles are also going to be what forms the cell plate, the beginning of the new cell wall. So after cytokinesis, we end up with two daughter cells that each contain all of the components of the complete cell. They have their own set of DNA and they have their own copies of organelles and everything that they need to be a complete functioning cell. Chromosomes are precisely distributed. Each new daughter cell has exact copies of the DNA. And it's noted that the orientation of cell division is important to the development. So kind of like which direction the cell is facing when it divides. But there's not really any apparent precise mechanism for distribution. This is something that scientists are still looking into, and they don't have a solid answer on how exactly the cell is able to evenly distribute all the components and all the cytoplasm to each new daughter cell. It happens, but it's one of those things that we're still not super sure about. So the tricky part about the cell cycle is how exactly the cell knows how to move into the next stage of the cell cycle or the next step of the cell cycle. Okay, the way that this is done is through this trigger substance that are protein kinases. Okay, specifically, they're cyclin-dependent kinases. Okay, what these CDKs do is they catalyze the phosphorylation of proteins that regulate the cell cycle, and they're activated by binding to cyclin. This is going to expose the active site because of this allosteric regulation. So as you can see, when that cyclin binds to the CDK, it opens up the active site, which activates that cyclin-dependent kinase. Once this happens, now my complex of my cyclin and CDK is able to phosphorylate these proteins that regulate the cell cycle. Now, these CDKs function at cell cycle checkpoints. So there are 4 checkpoints in eukaryotic division. The G1 checkpoint is triggered by DNA damage. So what we're checking here is to make sure that the DNA is all good and ready to be duplicated in the S phase. Okay, the S phase also has a checkpoint. This is going to be looking for incomplete replication or DNA damage. So as the S phase progresses and we make a copy of DNA, it makes sure that the copies that are made are complete and accurate. The G2 checkpoint is going to be checking for DNA damage. So is it ready to be to go to mitosis and be put into new daughter cells? The M checkpoint is going to be checking for chromosomes that are not correctly attached to the spindle. So if they're lined up properly along the metaphase plate, and if they are correctly attached to those mitotic spindles. Each CDK for each of these steps has a cyclin that is specific to it. Remember, these are proteins and their enzymes that are specific to the substrates that they work on. So the same concept applies here. Each CDK has its own specific cyclin that activates it, and it's only made at the right time. After that CDK acts and it does its job and progresses a cell to the next stage of the cell cycle, that cyclin is then broken down by a protease, which is a special enzyme that breaks down the cyclin. As this cyclin is broken down, this is going to now deactivate the CDK because it needs the cyclin to be active. So the whole picture is that this synthesis and breakdown of cyclins is what controls the cell cycle. This is what moves things on to the next stage. Okay, these cyclins are synthesized in response to various signals such as growth factors. Okay, if the cell is not ready to divide or not ready or able to move into the next stage, then those cyclins aren't going to be produced. So because those cyclins are synthesized and degraded throughout the cell cycle, you can actually see a fluctuation in the concentration of what we call the MPF, or the maturation promotion factor, or the mitotic promotion factor. So what this does is, as the cyclin is abundant, that cyclin will bind to the CDK. That cyclin and CDK complex is what we call the MPF. Okay, that is the maturation promotion factor. That's going to be what signals for the cell to continue on to the next phase. Once it continues to the next phase and it's good to go, that cyclin is going to be degraded. It's going to break down until it slowly accumulates for the next round of division.